This is Lecture 7, Psychophysiology Methods, and it corresponds to Chapter 7 of your textbook. Reminders and announcements. Everyone did a really nice job on Forums 4 and 5. I was really tempted to jump in and address some questions or just join the conversations, but I promised at the beginning of the semester that I was going to leave the forums to you guys, so I refrained. But I was particularly impressed when students were able to answer other students' questions, and there was some just really interesting back and forth conversations going on, so really nice work. Kind of inspired by the nice job that you did on Forums 4 and 5. Forums 7 and 8 are going to be structured similarly. Please do be careful to read the instructions. So similar to 4 and 5, in Forum 7, you're going to be making one post, and then in Forum 8, you're going to be responding to 2 posts. So make sure you respond to 2 posts, otherwise you'll only get half credit for the forum. And then finally, I'm going to be releasing exam grades soon. I need to wait to make sure that anybody with extensions has finished the exam, and then I'll release those grades to everybody. Your recommended readings for this week are 2 New York Times articles that are related, and they're recent articles about the kind of controversy surrounding CTE, which is chronic traumatic encephalopathy. This is brain swelling and damage that occurs from concussions, and especially repeated concussions. And there's a lot of coverage about it right now, both in research and in popular news about, because a lot of research is finding that a lot of players, especially in the NFL, suffer these repeated concussions and have like long-term brain injury that affects their behavior and their kind of emotional control as well. And so the first article is about Aaron Hernandez, who many of you are probably familiar with. I don't follow a lot of sports, but even I've heard of him. He was convicted of murder a while ago and died by suicide in his prison cell while he was, I think, undergoing an appeal for one of his murder charges. So they've recently released some of the findings of, they've examined Mr. Hernandez's brain, and I think he was only 27 when he died, but his brain was severely injured. He had severe CTE, especially in frontal areas, which, if you remember, is associated with impulse control, decision making, and as well in medial temporal structures, including the amygdala, and I believe the hippocampus was, or both hippocampi were damaged as well. So it kind of suggests that it's possible that some of the repeated concussions during his football career affected his brain in such a way that his behavior might have been affected as well. Now, I don't think they're trying to say that that his brain caused his history of aggressive behavior, because it seems maybe he kind of was aggressive and slightly violent anyway, but this might have exacerbated some of these situations. And the second article is a more general article about just widespread brain damage in NFL players. This research study had a sample of, I think, over 100 NFL players and found CTE in many of them, making their brains kind of look a lot older and more damaged than you would expect for people their ages. And I think it's this article that also notes that suicide and depression and a lot of psychological issues are more prevalent in retired NFL players, and research is examining right now whether there might be a link between CTE and concussions and some of these emotional issues later in life.
Okay, so jumping into Chapter 7, which is the methods chapter, and Section 1, study of brain structure and functions in general, I'm going to go through these methods a lot more quickly than your book did, and I will not ask you anything on the test that isn't in my slides for this chapter. So don't worry about getting too detail-oriented about the information in the book. Okay, so neuropsychological testing, these are tasks that are done typically with a psychologist or a neuropsychologist to measure cognitive abilities. So for example, Damage to the temporal lobes would typically cause memory impairments, and we have a whole battery of memory tests that are associated with different specific functions in memory. So you might do a test with them if you suspect that their spatial memory is impaired. You might do this block tapping test where the experimenter taps these blocks in a specific pattern, and the individual who's being tested, who can't see the numbers on those blocks, would have to tap it in the same pattern. So they would have to remember the spatial sequence. If you like, there's this group of neuropsychological tests that you can take online. And a quick note, these are not diagnostic, so if you don't do well on one, it doesn't mean you have necessarily a deficit in that particular cognitive ability. Any actual diagnostic testing must be done by a trained psychologist, and it must be interpreted by a trained psychologist or neuropsychologist. So lesion studies were one of the first ways that we started to understand that the brain and behavior are connected. A lesion is damage to any part of the brain, so this might be from a tumor, from stroke, from something being lodged in the brain, from traumatic brain injury. In humans, we observe the correlation between areas of the brain that have been damaged in individuals and how they act after the damage happened as compared to before the damage happened. In animals, it's a little more experimental, so scientists can do brain surgery on a rat, for example. If they want to know what the memory functions of the temporal lobe are, they might cut those parts, do brain surgery and cut the parts of the temporal lobe out of the rat, and then have it perform memory tasks later to see how that affects their memory functioning. As I was saying before, many of the earliest links that we made between brain and behavior were learned from post-mortem lesion studies. Because we did not have these neuroimaging techniques previously, the brains were examined after a patient died. And one caveat, over time, other parts of the brain will start to compensate for the injured area. So as we know from the previous section on developmental brains, our brains have some amount of plasticity even into adulthood. And if it's damaged, the brain can other parts of the brain can sort of absorb the functions of the damaged part of the brain. So an example of the importance of lesion studies, there's an area of the brain in the frontal lobe that we call Broca's area now, and this is because Paul Broca, a scientist back in 1861, had a patient who we know as Tan, and this patient had damage to the interior frontal gyrus on the left side of the brain. And the individual could only say the word tan after this lesion. So he would just say tan, 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 as if he was saying something, but only this one word was coming out. This led to the conclusion that this area of the brain was necessary for fluent speech production, as this was the ability that Tan lost after this brain part was lesioned. And this was backed up by other patient studies as well. Tan was just the first one and the most famous one. So this area is now called Broca's area. Broca's aphasia is the name now for what Tan was experiencing, or expressive aphasia is another name. And if you're interested, there's a YouTube video of an individual with Broca's aphasia. Similarly, Wernicke's area, another language area, was discovered this way. So Carl Wernicke in 1874 had a patient, and he noticed that had a patient with a lesion in the superior temporal gyrus, and this patient was able to create fluent speech, unlike Tan, but the content of the speech was absolute nonsense. So the patient was saying a lot of words, and complete sentences, correct grammar, but the actual meaning of the sentence was complete nonsense. And from this, Wernicke determined that this area of the brain, the superior temporal gyrus, was necessary for creating meaningful content in speech. And if you would like, there's a video of a patient with Wernicke's aphasia. Okay, another and slightly newer way of studying the brain is to actually directly stimulate the brain. This is called deep brain stimulation. an electrode is implanted deep in the brain tissue, and it stimulates specific areas to excite or inhibit neurons in those areas based on what the scientist or the physician wants to do. This has been used mostly therapeutically. It's a little bit too dangerous to use for just research. So it's been mostly used just therapeutically to improve Parkinsonian tremors, so going into the middle part of the brain to try to stimulate the brain and decrease Parkinsonian tremors. It's also been used to stimulate part of the brain believed to be kind of under-functioning in depression, and there has been some success in clinical trials of deep brain stimulation, relieving some people's depression. Of course, we can study the brain through drug manipulation. So this is administering drugs, targeting a specific neurotransmitter system, and then observing the effects, either through brain imaging or the behavioral effects. So one kind of, to me, a surprising thing that we used to do when I worked at a psychiatric hospital as a researcher was an amphetamine challenge. So this was a study looking at how amphetamine, which is a dopamine agonist, affects schizophrenia patients, and you might expect that it would affect schizophrenia patients quite strongly because overtransmission of dopamine is associated with schizophrenia, overtransmission in certain parts of the brain. And so amphetamine challenges, they would administer these amphetamines and then see how the brains of people with schizophrenia react differently to large doses, or to doses of amphetamines versus people without schizophrenia. So there are several different ways to measure electrical activity in the brain. There's of course single cell recordings where you record just one cell and the action potentials, changes in membrane potential within that single cell. We talked about that earlier in some of the earlier chapters. And then there's electroencephalography, or EEG. And EEG is just looking at kind of bigger spatial areas of the brain and electrical activity within that. So you've probably seen the caps that people wear with the little electrodes implanted within those, and each electrode is recording the electrical activity of all the neurons below that part of the skull. There are two main ways that people use EEG. One of them is looking at event-related potentials. So an individual has the cap on and is shown a set of stimuli, and each of those stimuli is like an event, and they measure brain activity that corresponds to that event. So how the brain responds to, for example, like letters on a screen, something like this. And then if you look in the graphic on the top right, you can see that there's different segments of EEG. Because the changes in brain electrical activity are so small, you have to do many trials of the stimulus and kind of average them all together, and then this creates this averaged waveform. And it's very quick, very good temporal resolution, meaning we can tell when things happen very close to the time period that they happened. So you can see down here this graph of the averaged ERP waveform. These events can happen in under 100 milliseconds, so we can measure something called the P1 component of a waveform that happens 100 seconds after an event occurs. And all of these waveforms have been pretty well researched and correlate with different cognitive events. For example, I used to do research with the P3 component down here. It says it's at about 400 milliseconds. It's actually a little bit closer to 300 milliseconds. That's why it's called P300 or P3. And this is associated with what we call oddball stimuli. So whenever somebody sees an unexpected stimulus, for example, if they're looking at a stream of letters and suddenly they see a number, that's going to evoke this P300 or P3 ERP waveform component in the brain. There's another type of analysis of EEG that's a little more recent. It's called time frequency analysis, and this records changes in synchronized neural activity in the brain. So sometimes brain areas create action potentials. All the neurons in the brain areas create action potentials together, and that's called actually when you talk about brain waves, that's more what people are talking about. So synchronized neural activity. And this is measured in frequency bands. So down here in the green at the bottom, don't worry too much about the details of this graph because it's actually a rather complicated technique. But if you see down here the frequency in hertz, and there's 5, 10, 15, 20, so these are, this is measuring different bands of neural activity at different frequency levels. So at 5 hertz, this means that the neurons are firing 5 times within 1 second, for example. So all these neurons are firing together 5 times every 1 second. And that is useful for things like sleep and changes in consciousness, changes in attention, things like that. Okay, moving on to anatomical techniques. Anatomical magnetic resonance imaging, or anatomical MRI, takes high-resolution images of the brain, and it does this by measuring differences in the hydrogen content between different materials in the brain. You don't need to know the specifics of this unless you take further courses, so I'm going to go over it pretty briefly, but essentially what you want to know is when you look at an anatomical MRI image, more dense materials are a lighter color, and the less dense materials are a darker color. So if you look down at this image on the bottom right, you can see that the skull, for instance, is very bright white, and that's one of the more dense materials in this image. As well, it's important to know that often in research and also medically, images are mirrored, so there's a left-right reversal. When you think you're looking at the left, you're looking at the right. When you think you're looking at the right, you're looking at the left. So you see a little R and L in these images, and that will tell you what side you're actually looking at. MRI machines in general, if you've never been in an MRI, it's important to know that you can't have metal materials or ferrous materials, like a specific type of metal, in an MRI facility. People who have done research with MRI were all a little bit obsessed with what happens when you accidentally do bring ferrous materials into an MRI machine, and there's a lot of safety videos on the internet of people who do that. So if you're interested, there's one where they smash a watermelon with a gurney or something like that against an MRI magnet, because the MRI magnet is an extremely strong magnet. So even if you have something like a safety pin in your pocket, it can injure somebody who's standing between the safety pin and the machine. MRIs take a lot of pictures, so like slices of the brain, sort of take pictures of each slice of the brain, left to right and then up to down, and this creates voxels, which are like 3D versions of pixels of the brain. So it's anatomical MRI is very good spatial resolution, the best spatial resolution of the methods that we're talking about today. And then there's computed tomography, which is CT, like a CT scan. This uses X-ray, so it's rather different from MRI. X-ray absorption varies based on different tissue density in the brain, which creates this image. CTs are good for finding tumors and lesions. They're less expensive than anatomical MRI, but there is less distinction, less spatial resolution, and less distinction between white and gray matter. DTI, or diffusion tensor imaging, also uses an MRI machine, but this is measuring movement of water molecules through white matter tracks. So it's showing connectivity between different areas of the brain. And then these images, you use a program to color the images so that it's easier to visualize where the different tracks are going. Otherwise, it would just be kind of a muddled, it would be really challenging to see where the different tracks, which tracks are connected to each other. Functional magnetic resonance imaging, or fMRI, you've already seen a number of these types of images, and this is essentially, again, you use the MRI scanner, and it measures which areas of the brain are engaged in a task. So this actually shows you functioning of the brain rather than just structure. So MRI actually makes its images by measuring differences in the oxygen levels of the brain in different areas of the brain. So the comparison of oxygenated versus deoxygenated blood, More oxygen in an area means that area is more likely to be engaged in a task, and it's kind of it's a little bit complicated why that is, so I won't go into it, but it's sort of the source of some controversy as to whether we're actually measuring function of that area, or and how good the actual spatial resolution of functional MRI is. However, in general, fMRI has good spatial resolution and poor temporal resolution. So we have a good idea of where the activity is happening in general, in which brain area, but there's a lapse of like 2 to 3 seconds between when the area is being used and when this oxygenated blood shows up. And so it's not time locked very well to the task that the person is doing while they're in the MRI scanner. And most of you already know this, but if you look on the top right, you can, the areas, the colors indicate level of activation, and so the brighter the color, the whiter or lighter the color, the more activation in that brain area. And then going to this darker red means slightly less activation in that brain area. There's one con of fMRI is that there's a high likelihood for false positives. So false positive means finding what we think is significant activity in an area of the brain when there actually isn't. And this is because the brain, in part, it's because the brain is sort of active all the time, and so you have to compare the activity against like a baseline or a resting state. There's also a statistical reason called multiple comparison correction that I won't go into, but people complain about that a lot for fMRI, that you're doing so many comparisons, like so many statistical tests, that there's a higher likelihood that you're going to find false positives. If you have taken statistics, maybe you remember why that is. There's this famous story of the dead salmon where when you're testing your MRI equipment, sometimes you use what's called a mask, or you just put sort of an object that you can see inside. So often, well, inside the image, inside the scanner, so some people use like a watermelon. This one lab used a dead salmon to test the scanner and their program before they used it for participants. And just by chance, because of this high likelihood for false positives, activity showed up in the brain of the dead salmon. So you can see over here, this is a picture of that dead salmon. So this red activity showed up, and it just happened to be in the brain region of the dead salmon. And a lot of people took this as an illustration of the high likelihood for false positives of fMRI research, and that we should be careful when we interpret fMRI research. In general, you can trust MRI research. Many of these studies have been done over and over again, and the same results have been found, as one should always do in good science. But it's always good to keep in mind the drawbacks of any imaging technique. So then there's PET, and this is used to study metabolic activity in brain cells. Water with radioactive molecules are injected into the person's blood, and PET is useful because it can detect neurotransmitter activity, the density of receptors for neurotransmitters, and other things that functional MRI cannot. Functional MRI just looks at differences in oxygenated blood. A drawback of PET is that it has pretty poor spatial resolution compared to fMRI. And also you have to inject people with radioactive molecules, which isn't dangerous, but some people don't like to do that, especially for research. In imaging, it's become more and more common to combine techniques. So as you probably picked up throughout this section, every method has its strengths and its weaknesses, and combining techniques can combine strengths. So a really great combined method is EEG plus fMRI or structural MRI. So EEG has good temporal resolution, poor spatial resolution. fMRI has really good spatial resolution and poor temporal resolution. So you add them together and you get good spatial and temporal resolution. Another thing people will often do is look at people who have lesions in their brain with any kind of brain injury, imaging to see if there are functional and structural differences. Neuropsychology or like cognitive tasks can be used along with brain imaging to see if there's differences in the size or thickness of the cortex for people with different cognitive abilities, and many anymore.